Hi everyone, welcome to part 1 of topic 11, and in this part we're going to be talking about DNA, chromosomes and genes. Now the detailed learning objectives are to be able to define and describe chromosomes and karyotype, be able to describe the processes that result in gene expression, be able to discuss the distribution of chromosomes to offspring, be able to explain what the genetic code is and what it is coding for, be able to define genetics and explain how chromosomes are related to inheritance, and be able to differentiate between the following sets of terms. So dominant and recessive, genotype and phenotype, and homozygous and heterozygous. Now, just a little bit of an introduction to genes. And remember that the development of a person is determined by the gene-bearing chromosomes that it inherits from its parents through conception. So through the fertilization of the secondary oocyte and the sperm cell. Now, genes are these discrete DNA segments that contain the code or the recipe or the blueprint for protein synthesis. And when we say that a gene is expressed, this leads to an observable characteristic or trait. This might be hair color, might be the presence of freckles, it might be determining sex, the ability to roll your tongue, and your blood type, to name just a few. It's a little bit more complicated than all of that, though. A gene's ability to promote the development of a specific trait may be enhanced or inhibited by interactions with other genes, and as well as that, environmental factors that we'll talk about later. And all of the human genes combined together then are known as the human genome. Now, just to be sure that everybody understands, we are not talking about this type of genes. We're talking about this more boring little picture here. So before we get into the actual accessible content, just some more general information about the Human Genome Project. Now, this was completed way back in 2003. And the sequence of the entire human genome involved sequencing 3 billion nucleotide bases. So adenine, thymine, guanine, and cytosine. It is an integral tool, or it has been an integral tool in the identification of 1,800 disease-causing genes. And it has been used to develop over 2,000 genetic tests for disease-causing conditions. It's been sort of instrumental in developing the technology to allow the mass sequencing of lots of different things. And I've just listed a few here. So other organisms, human populations, cancer samples to identify causative agents and direct personalized therapy for individuals with cancer. And it continues to facilitate advancement of new technologies like genome editing as well. And you may be wondering, what is genome editing? Well, very simply put, DNA is either inserted or it's deleted or it's modified or it's replaced in the genome of a living organism. And technologies such as CRISPR-Cas9 are being used to do this. Now, to understand gene expression, we just need to revise a little bit about deoxyribonucleic acid, or DNA. It's a type of organic molecule and it stores hereditary information. It's, believe it or not, it's like 99.9% similar between individuals. And of course, identical twins will have the same DNA. Most cells in the body contain the same DNA. It itself is very long. It's a 2-stranded polymer that stores information using a code determined by the arrangement of 4 different monomer or nucleotide bases called adenine, cytosine, guanine, and thymine. And DNA itself can act as a template, either for making copies of itself or to be decoded. So it has recipe or instructions to be decoded into functional elements. Now, each cell contains around 2 meters of DNA. And so that's a lot, right? So to prevent this from becoming a tangled mess, it needs to be effectively packaged. And part of this packaging involves being organized into small discrete pieces called chromosomes. Now, normally these are not visible, but when cells divide, which you can clearly see here through this really cool animation, the DNA in the chromosomes condenses and it can be visualized under a microscope, so only in dividing cells only. Now, for all of you that are wildly into all of this, there's a really cool video here. It's called DNA to Chromosome. It's an Australian video, so have a look at that in your own time. Now, in terms of the definition of a chromosome, it's a thread-like structure which is located inside the cellular nucleus. Each chromosome then is made up of proteins called histones, and they're kind of providing the support, and it's around this histone that this DNA can kind of tightly coil. And so it does contain a single molecule of DNA, and DNA then is passed on to offspring, and it contains the specific instructions that make each type of living creature unique. And so in terms of a definition of genetics, well, it's actually just the study of heredity. And through genetics, it helps us to understand why you might have blue eyes, but your brother or sister might have brown or green eyes, why you are tall, but they are shorter, while you might have freckles, they don't have freckles. But it's not just all about those kind of little things as well. Genetics helps us to understand how you might be predisposed to a particular illness or condition later in life, but your siblings might not be. So chromosomes contain the DNA that is inherited by offspring from the union of a sperm cell and a secondary oocyte during the process of fertilization. All right, so we're just going to have to revisit mitosis and meiosis just for a couple of slides. And meiosis, of course, is going to be important in genetic variability, but I'll get to that later. So in terms of mitosis, everybody, body cells divide to produce two identical daughter cells through this process. Remember that all of the genetic material, so the chromosomes, need to be replicated before the cell divides, so that each daughter cell gets the same number of chromosomes as the mother cell. And it is at this time that we can see the full complement of chromosomes that you're looking at over there. We have these different phases in the cell cycle. We have something called interphase, and that's the phase between division or mitosis. And then mitosis itself is split up into prophase, metaphase, anaphase, and telophase and cytokinesis. Okay, so down here we have interphase. We've got, our nuclear envelope present here. As we move through that first phase of prophase, that nuclear membrane disintegrates and releases those chromosomes. Remember that they're condensed here in prophase. And we also have these centrosomes that are migrating to the opposite end of the cell, and we getting this formation of this mitotic spindle. Then as we move through metaphase, remember we have sister chromatids, because those chromatids have been replicated to ensure that, the daughter cells get the correct amount of chromosomes. So they line up on this equator here via the centromere attached to that. And then what happens in anaphase is we can see the migration of one set of chromosomes towards one end of the cell, and the migration then of the other set towards the other end along these spindle fibers. And then throughout telophase and cytokinesis, we have the formation of a new cytoplasm. We have this cleavage furrow forming that's going to, get deeper and deeper until it eventually splits off the two daughter cells, and we have this new nuclear envelope forming there. And so the whole process of mitosis then ensures that there's, genetic continuity. As mother cells divide to form daughter cells, they are identical to the mother cell. So meiosis then is a specific type of cell division that only occurs in the development of the gamete, so the secondary oocyte and the sperm cell. And it involves a process where only one of each pair of chromosomes is packaged into each sperm or secondary oocyte. So there will only be 23 chromosomes in each of the gametes, so we say it has the haploid number of chromosomes. Then when there's union of the sperm cell and the secondary oocyte during fertilization, a full set of 23 pairs is restored, or 46 chromosomes being that diploid number, or 2n. So down here we have meiosis 1 and we have meiosis 2. And meiosis 2 is very similar to mitosis, right? But in meiosis 1, it separates the homologous chromosomes, right? So remember, just prior to meiosis, you're going to have the chromosomes duplicating, and they form these sister chromatids, right? So we have in prophase 1, we have the homologous chromosome pairs, and we get this exchange of segments, okay? They then kind of line up in metaphase on this equator. So we get this formation of these tetrads that line up. During then anaphase, the sister chromatids remain attached. We have the homologous chromosomes separating, and that results kind of in the pairs of homologous chromosomes splitting up. And as a result of that, then we get two haploid cells forming, but the chromosomes are still double, right? Because all that genetic information was replicated before meiosis began. Meiosis 2 then just involves the separation of these sister chromatids. So again, it's very like mitosis. You can see these, these centrosomes migrating formation there of the mitotic spindle. We have the sister chromatids then, lining up. And then in anaphase, we have the sister chromatids separating. And as a result of that, then in telophase and cytokinesis, we have the haploid daughter cells forming. So during another round of cell division, which is this meiosis 2, the sister chromatids finally separate, resulting in the 4 haploid daughter cells containing single chromosomes. Now, I'll get back to quite a bit of this in a few slides' time when we're talking about how genetic variation occurs. Now, for those of you that have never done any of this before, let's just go back a step, right? So we talked about DNA. We have inside cells 1 meter of DNA that's from the mother's side and 1 meter of DNA or paternal DNA from the father's side. Now, if we then just decide we're going to cut each of these into 23 smaller pieces, you get something like this, okay? Now let's actually place all of these pieces side by side. So from the mother and from the father, okay? And now let's actually take some of these like so and place them side by side here. We're now going to go and look on and show the result of doing this for all of the rest of pieces of the DNA that came from the maternal side or the mum and the paternal from the father. And what you end up with is with something like this. So each bar is a chromosome. And when we combine, these two kind of chromosomes, we call them a homologous pair. And in humans, there are 23 pairs, so 46 chromosomes. And for each pair, 1/2 comes from the mother and 1/2 comes from the father. And some of these pairs of chromosomes are larger than others. And we'll have a look at that on the next slide. So what you're looking at here are all of the homologous pairs of chromosomes from the largest down to the smallest here. And we've also got this pair of sex chromosomes here. So this image is just showing you an individual's complete set of chromosomes, and they're arranged into something that's known as a karyotype. And a karyotype is just a display of a person's chromosomes, and it's used to look for either abnormalities in chromosome number, or it might be in chromosome structure. So there might be like deletions on some of these chromosomes or reassortment of genetic information. As I mentioned, one pair of chromosomes are the sex chromosomes, and the sex chromosomes are the chromosomes X and Y. Here you can see a karyotype for a male, because there is one X chromosome and one Y chromosome. If there were 2 X's, it would be female. So these sex chromosomes determine a person's genetic sex, right? And the other 22 pairs of chromosomes are called autosomal chromosomes. So each one of these chromosomes there are autosomes. And so just a little bit more about the karyotype here. It is a display or a description of a person's complete set of chromosomes, so how many they have, and also the different sort of banding patterns that are occurring. And by examining this display of chromosomes, things like Down syndrome, for example, can be detected, which you're looking at here, where there is an extra chromosome 21, and that's why it's called trisomic 21. Now, this can occur during, an error in meiosis, where there is something called a non-disjunction event occurring, where one of the gametes then ends up with an extra chromosome, so that the fertilized egg then has 47 chromosomes instead of 46. Genes then are the functional units of heredity and responsible for the transmission of different genetic traits from parents to offspring. Each gene, as you can see here, is a small region of DNA and a chromosome that contains information which can be decoded then to make protein. Now, you don't need to know the information here, but this is just showing you the X chromosome, and it's showing you different sort of locations of genes that are involved in disease. And the X chromosome itself has about 800 genes. Proteins, remember, specify structure and function, and therefore overall body function and homeostasis. And a gene also contains regulatory information that dictates when and where a protein should be produced. So now, looking at protein synthesis from, gene expression, we do have to revisit these two steps of transcription and translation. Remember that the genetic code contained in DNA uses those 4 nucleotide bases, being adenine, cytosine, guanine, and thymine. And this code then is converted into protein via a 2-step process, which you can look at if you click on this link, or you can look at directly through this little video here. Now, initially, the genetic code contained in DNA in those triplets of nucleotides is transcribed into an RNA molecule. And this occurs in the nucleus, and it's called transcription. Now, the RNA then is carried in sets of 3 nucleotides as well, and it uses the 4 nucleotide bases adenine, guanine, cytosine, and uracil. And these combinations of 3 of these nucleotides or letters are called codons. So each codon will specify an amino acid during translation, which is the formation or the synthesis of a protein according to the sequence in the messenger RNA molecule. For example, GAU specifies the amino acid aspartic acid, and CGA for arginine. This RNA, this messenger RNA molecule, leaves the nucleus and it's translated into protein by ribosomes. And this whole process then requires the information in the messenger RNA molecule. It also involves another RNA molecule called transfer RNA and another one called ribosomal RNA that forms the ribosome. We're not going to go into that in detail here, but the proteins then make up the structural and functional components of cells. And just to summarize all of this, the genetic codes that are stored in the DNA of genes instruct cells on which proteins to make them, when to make them, and how much of them to make them, and where in the body to make them. A very simple example here is insulin is a protein hormone, and it's only made by specialized cells within the pancreas. And that's because these are the only cells in the body to express the gene that encodes insulin. And then we know, of course, that the amount of insulin produced partly depends upon blood glucose levels. When blood glucose levels are high, the gene encoding insulin gets switched on or turned on more often, and more insulin is made. Now, there are about 25,000 genes that make up the human genome. And most genes are present as 2 copies, one on each chromosome. So here you can see a pair of homologous chromosomes, and we've just put in here genes on these chromosomes. So each copy then is referred to as an allele. So each copy of the gene is referred to as an allele. And in most cases, they are the same and perform the same function, but in some cases, they are different and perform different functions. For example, in the generation of skin color or hair color. So if we go over here, we can see that at this locus, which is just a particular spot on a gene, we have the presence of the same allele. okay? And in this situation, we call this homozygous. So homozygous is the same allele of a particular gene present on each of the chromosomes in a pair, right? If we go down here, you can see that we have something called heterozygous. And heterozygous means that there are different alleles of a particular gene present on each of the chromosomes in a pair. So on this slide, we have some important terminology referring to alleles. So alleles are specific forms of a gene. They're found at the same locus on homologous chromosomes. When they are homozygous, it means that there is inheritance of two identical alleles for that particular trait. And when they are heterozygous, there's inheritance of two different alleles for a particular trait. Now, we also have these terms dominant and recessive. And now a dominant allele is one that masks or suppresses the expression of another allele. So when an allele is dominant, it's the presence of that allele which will determine what the appearance of that trait is. Recessive then is where an allele whose expression is masked or suppressed by a dominant allele. So for this to be expressed, then you need to have two copies of these recessive alleles. So one from the mother and one from the father. Now, dominant alleles are represented using a capital letter. So it might be capital T or capital A. And then a recessive allele is represented by using a lowercase letter. So a lowercase t or a lowercase a or other ones. Other important terminology to know about is the difference between genotype and phenotype. Genotype, as its name suggests, describes genetic makeup. So it's actually describing the combination of alleles that a person inherits from their parents. For example, it might be heterozygous, so a pair of alleles that are different, right, for a particular trait. Or it might be the inheritance of two similar alleles, but they might be two dominant alleles, or it might be two recessive alleles for a particular trait. And as I mentioned, it is common practice to use letters that look different when capitalized versus lowercase. Phenotype then describes what is being expressed, all right? So the way that the genotype is expressed. So it describes the characteristics of something, all right? Easiest way to look at it here is looking at the phenotype. So what somebody looks like for a particular trait. Now you can see here the phenotype here is red hair, and it's determined by a single pair of recessive alleles. So if we say H for hair, we would have two lowercase h's to describe that red hair there, okay? The phenotype here you can see is freckles as well. It's an example of strict dominance, and it requires only one allele for the trait to be expressed. So if we take the trait for freckles being F for freckle, you only need one dominant allele for the expression of that trait, and you can see it's being expressed here. So this person here might have, be homozygous dominant, so have FF in capitals, or could be heterozygous, so just have one copy of the dominant allele being the capital F and one smaller recessive allele. But remember that the dominant allele in this case masks the effect of the recessive allele. Okay, so we're now going to identify two important sources of genetic variation that occur due to meiosis, right? We get the deal here. We know that meiosis occurs in two divisions. Here's meiosis 1, where we get, the halving of the number of chromosomes. And then we have meiosis 2, where we have to split those sister chromatids, right? So what happens is genetic variation can occur. So one cell starts off with 23 pairs of chromosomes, right? So those 23 of those are from the mother and 23 then from the father, right? Now, the cell then copies all of this DNA, right? Because you need to have it copied for the division to occur. So what happens now is there's 23 original chromosomes from the mother and 23 copies. and also 23 original ones from the father and 23 copies, okay? Then through the process of, metaphase, and what we get is reassortment of some of this genetic material. So you can see we're getting swapping of pieces of material here between these chromosomes. Okay, so that some elements of the father's DNA is getting swapped for bits of the mother's there. And this follows through very simply here, and you can see then that at the final product of meiosis, we have those 4 haploid cells, and they can contain, variations of genetic material. So that one might be all, from the mother, one from the father, and then we can have these all, different variations there. And so the 2 main sources then of genetic variation are recombination or crossing over. And that's what you're looking here. So the homologous chromosomes can exchange DNA. So here we have our homologous chromosomes. Remember, they're going to consist of 4 sister chromatids. And we get this crossing over here when these tetrads are formed, where some of the chromosomal information is exchanged, and this occurs obviously in the formation of the sperm cells and secondary oocytes. The other thing that can occur is this independent assortment that occurs again during meiosis as the sperm cells and oocytes are formed. Paternal and maternal chromosomes get mixed up and redistribute independently of the other chromosome pairs. And that's what you're looking at here, as denoted by the red and the blue. So again, by the time you get the generation of these haploid cells, there is variation. There can be variation in them. There can also be an effect of the environment as well on gene expression. So certain components that are found in food, for example, mercury, or cigarette smoke, or drugs, be they legal or illegal drugs, alcohol as well, and radiation such as x-rays, along with all sorts of other factors, can cause mutations in the DNA of the developing embryo or fetus, and that can, in some cases, then lead to congenital defects. There can also be effects on gene expression after birth as well. So all of this happening here in utero as the embryo and the fetus is developing. But after birth as well, gene expression can be impacted too. So poor nutrition can affect brain growth, body development, and height as well. And I think height is a really easy one to sort of understand if you kind of go back in history, where our nutrition was not as good as it is today. People tended to be much shorter compared to today. And childhood hormonal deficits can lead to abnormal skeletal growth as well. I will add here, in terms of the height, obviously there is a genetic component to it. I've had very good nutrition, but I never really grew very tall linearly. So yes, I am vertically challenged.